Welcome to Genome Journeys. Today, we're diving deep into the world of our DNA, exploring tiny entities that are shaking up our understanding of evolution. Ever heard of jumping genes? Well, meet their even more intriguing cousins, introners. These aren't just passive bits of genetic code. Introners are selfish genetic elements, almost like molecular stowaways, with a remarkable superpower. They can create new introns, those non-coding sequences, right inside our genes. Think of them as editors constantly adding new notes in the margins of our genetic blueprint. But here's where it gets truly mind-bending. Scientists recently discovered that these introners aren't just moving around within a single species. Oh no, they're performing incredible leaps between completely unrelated species. Imagine an introner from a sea sponge suddenly appearing in a marine protist called a dinoflagellate. We're talking about organisms whose last common ancestor lived over 1 billion years ago. How is this possible? The leading theory points to viruses, particularly giant viruses, acting as unwitting interspecies taxis, ferrying these introners across vast evolutionary divides. This horizontal gene transfer, as it's called, means introners can shuttle genetic material between lineages that would otherwise never mix. So what happens when an introner sets up shop within a species' genome? They can trigger what's called an intron burst, where a genome rapidly gains many new introns. This isn't just random chaos. By inserting themselves, introners can subtly influence how genes are expressed, and, crucially, they can enable something called alternative splicing. This is a fantastic cellular trick where one gene can produce multiple different proteins, dramatically increasing an organism's functional toolkit. It's like having a single recipe that can be tweaked to make several different dishes. Now, you might wonder where these adventurous introners hang out. They're quite widespread, found in algae, fungi, various single-celled organisms, and even some marine invertebrates like sea urchins. Interestingly, they seem to prefer aquatic life. This makes sense, as horizontal gene transfer, that species hopping ability is generally more common in marine environments. These introners themselves are believed to have evolved multiple times from different families of transposable elements. Their survival strategy is clever. They need to be efficiently spliced out of the gene's important coding sequence so they don't harm their host, ensuring they can keep on copying themselves. And here's a fascinating thought. Could introners actually help create new species? It's an active area of research. If different populations of a species start collecting unique sets of introners, it could eventually lead to them becoming reproductively isolated. Imagine two groups speaking slightly different genetic dialects due to these introners, making it harder for them to produce viable offspring together. We've already seen that distinct lineages, like the green alga asterisk micromonas, have their own unique families of introners, hinting at this connection. So these tiny introners, once dismissed as mere genetic clutter, are emerging as powerful agents of change. They generate new introns, boost genetic complexity through alternative splicing, and even bridge evolutionary chasms with horizontal gene transfer. They're a perfect example of how seemingly selfish elements can inadvertently become major drivers of innovation and the beautiful diversity of life on Earth. Thanks for joining us on Genome Journeys. Until next time, keep exploring the wonders within.